welcome to biology, eukaryotic cells, eukaryotes. This is a continuation of the previous video about eukaryotes versus prokaryotes, and we need to look at the real structure of an eukaryotic cell, depending on what is expected from us from IB. So this is IB chapter 1.2, that's the unit. And in this breakdown, we are talking about the eukaryotic cell. We are going to talk about the real examples of eukaryotic cells, in particular a plant cell and an animal cell. And then we look at the organelles structure related to function, which is the adaptation of each cell. So this is going to be of very great importance. Like as usual, you can pause, take notes. You can pause, read again, listen as you take your notes as we move. So we shall have some assignments done, as well as some simple quizzes. And after this unit, we are going to have questions and answers, multiple choice, and short answer questions for chapter 1.1 and 1.2. So I guess you continue to be on this and then we move. Thank you. So what do we call eukaryotic cells? We said eukaryotic cells, they are basically cells which were formed after the formation of the nucleus or evolution of the nucleus. So whenever you see a cell with a specific nucleus, it is referred to as the eukaryotic cell. So eukaryotic cells have compartmentalized structure. As you see, this is one compartment, another compartment, and other regions. So different regions containing different activities. So that makes what we call compartmentalization. Unlike prokaryotic cells, which lack compartmentalization. So that's another good definition or difference that marks the difference between the eukaryotic cell and the prokaryotic cell, those two domains. So with eukaryotes, as you hear the word eukaryotes, that is after ryotes, that is after formation of the nucleus. So eukaryotic cells are cells which have double membrane organelles. Double membrane organelles. Double membrane organelles or are cells which have compartmentalization when in their structure. Don't forget we are looking at the outer structure of these cells, the detailed structures. These structures can only be done or be seen using a sophisticated electron microscope that gives us what we call the electron micrographs, which we can look at and then we study the details. The other way we can have these organelles produced out, we get them from what we call centrifugation. A centrifuge is a device that breaks down a cell components in their respective units and then dyes them differently for easy visibility and study. So most of you will meet all these ones when you're doing your research at universities or during your masters. That is if you continue with biology to university and any other place. So why do we say, why should cells have compartments? That's the question we have. Eukaryotic cells are divided into compartments, which compartments are now referred to as organelles. Organelles, these are structures, subcellular structures that have specific functions. As organelles are subcellular structures that have specific functions within that cell. So when we ask you to define what an organelle is, they are subcellular structures that have specific functions they serve within that cell. So by petitions are known as membranes. They have many different compartments which are separated by double and single membranes, such as the nucleus. The nucleus is double membrane organelle. Mitochondria are double membrane organelles. Rough endoplasmic reticulum and endoplasmic reticular are double membrane organelles. So if you go and read page 21 in your textbook, you can make some summaries of the importances of compartmentalization. When you talk about the importances of compartmentalization, we are finding basically the importances of having different organelles are being located in these cells to serve different purposes. If it is a plant cell, they also have, most of them are similar. Most organelles are similar apart from one organelle which we call the chloroplast. So plant cells have a chloroplast, animal cells don't have a chloroplast. So that makes a difference. Another difference lies in the plasma membrane, whereby the plant cell has a cell wall, an animal cell doesn't have a cell wall. Then we can still continue and say, why do we need compartments? The importances, if you look at this, these are detailed structures of eukaryotic cells, and that is the plant cell and animal cell. I want us to take a good look at an animal cell. If you look at this animal cell, it has one membrane, which we call the plasma membrane, that passes around, that makes this boundary. And if you look at it inside, it has an organelle with dots that is surrounding the nucleus, not so close to the nucleus, but around the nucleus. Because in this, the nucleus is in the center. And in this one, the nucleus is on the side. It's not in the center. What is in the center in a plant cell? It is what we call the vacuole. And what is in the center in an animal cell is the nucleus. Now, if you continue to look at this, the nucleus is surrounded by a double membrane crystal like substance that is rough, that contains stuff on it. That contains stuff on it. For example, it has ribosomes, and also it has ribosomes. So that means this structure is called the rough endoplasmic reticulum. It's called the rough endoplasmic reticulum. Rough endoplasmic reticulum. And then from here, we have this sausage-like structure, which we call the mitochondrion. As it's one, it's a mitochondrion. When there are many, they are mitochondria. So this mitochondrion it's the powerhouse of the cell. Then when we come to these structures here, they are different from this. And then I also have the same structures in the same color, but in this case, they are different. They are not rough. They are different from the rough endoplasmic reticulum, which we call the smooth endoplasmic reticulum, same color. And then when you come here, we look at the cytoskeleton. And I learned this from my lecturer. He was called Remick. He used to talk much about the cytoskeleton. It's one of the things I didn't know when I went to university. So these ones make the cytoskeleton of the entire cell. And here we have the Golgi apparatus. which we call the Golgi body or Golgi apparatus, and also have the lysozymes. Lysozymes. So if you look at the animal cell, basically the following 5 features should not run out of your head. Start from the middle with the nucleus. Then on sides of the nucleus, we have the rough endoplasmic reticular. When they are many, they are reticular. Then after the rough endoplasmic reticular, you come next to the smooth endoplasmic reticular. Then from smooth endoplasmic reticular, we come to what we call the centrioles. We come to the Golgi apparatus and then the lysosomes. So when you come to an animal cell, plant cell, with a plant cell, we may have a few structures similar, but many are different. Both cells have the endoplasmic reticular. You can see, and the endoplasmic reticular is just surrounding the nucleus. The nucleus in this case is on the sides, and it's a rough endoplasmic reticulum, and that's the one that has ribosomes. Then the one next to it that comes without the ribosomes, it's what we call the smooth endoplasmic reticulum. Each one has a function. Then the cell vacuole. This is the vacuole. The cell vacuole is in a center and it's large. So a cell vacuole is large and is located in a center. This is the animal cell, plant cell. Then another distinguishing feature is the one where we have the smooth and where we have the chloroplast. A chloroplast is a pigment that is a structure, is a cell that produces chlorophyll, and it's responsible for the green coloring of the cell, and it's the site where the light dependent stages and light independent stages of photosynthesis take place. So this is so beautiful and it's so nice. So we can always proceed, and then we see, if you look at this same cell, it has a structure, it has a cell wall. This is a cell wall. One cell is connected to the other by the wall. So that's one of the importances of the 2 eukaryotic cells in details, the outer structures of the 2 cells. So checking for the different structures, then we can see, so the animal cell, algae, protozoa, fungi, and plant cells are examples of the eukaryotic cells. So if you look at one here, we shall see the different compartmentalization, and this is a micrograph. And because we see the nucleus, this side is on the side, we see the Golgi apparatus, they are not so visible, you need to be a little close. And then Golgi apparatus are mostly close to the lysosomes, as well as they are just close to the lysosomes and the vacuoles. Then from that we go to the mitochondrion, as you can see, a sausage-like structure. Then towards the end we see the rough endoplasmic reticulum and the plasma membrane outside. So this becomes, because the nucleus is so huge and we don't see the vacuole, this automatically becomes an animal cell. So what should we know about animal cells? 1, single membrane. Number 2, the large nucleus. And number 3, the mitochondrion on the sides. Then the endoplasmic reticulars. And then we look at the ribosomes as well, because the rough endoplasmic reticulum contains ribosomes. That's why it looks rough, like the skin of a jackfruit. So this shows the organelles of the liver. So this is just the liver cell. And when you analyze this liver cell, its compartmentalization is a little different. You see where the nucleus is, and then you see where we have the lysosomes. These lysosomes here, they are quite a number because these lysosomes they produce the other serious enzymes, which we call the hydrogen peroxide. And if you see, you can be observant, you see that there is a lot of mitochondria. So that means this is a very active cell. Presence of mitochondrions, it shows it's very active cell because there is a lot of aerobic respiration, and because there is a lot of the active, it's an active cell. So even the lysosomes that produce the peroxisomes enzymes, which produce hydrogen peroxide, which reduce the toxicity that is produced, is highly here. So that shows that this cell is an active cell due to the vast number of mitochondrions present. And that means because it's active, there is a lot of peroxisomes that are produced by the lysosome enzymes responsible for a detoxification of the human body, as you can observe and see. Then on the outer part of this cell, we have what we call the plasma membrane. A plasma membrane is one, it lacks what we call the cell wall. Therefore, we shall look at it in detail as we move. So eukaryotic cells are usually 5 to 100 micrometers big as compared to the prokaryotes, which are zero point 0.1 to zero point 0.5 micrometers. So this is 5 to 10. So they are bigger. As for starters, a 1 micrometer is equivalent to 0.001 millimeter. So for you to get 1 micrometer, you get 0.001 millimeter is equivalent to 1 micrometer, and then 1 nanometer is equivalent to 0.001 micrometers. 1 nanometer is equivalent to 0.001 micrometers. So a nucleus is membrane bound and have membrane bound organelles, as we've seen. And then we have the nucleus. Now let us look at structure, parts, its organelle related its function, adaptations of every organelle. And when I talk about adaptations, I'm basically talking about structure related to function. Structure related to function. The structure related to function. So, generally, if you look at this, the nucleus, because I'm starting from the center, the nucleus, if you are very observant, you'll see it's like a surrounding membrane. And oh, just a minute, doesn't have to happen, doesn't have to happen, doesn't have to happen, doesn't have to happen. Just a minute. Well, sorry about that. So, like we said, this nucleus has, if you check it out, it has, it's spherical in its structure, but inside it is another extra ball which we call the nucleolus. And then it's in the nucleolus where we find lots of the DNA and ribosomes and all that stuff. Then on the sides, we see what we call the membrane. If you look at this membrane, the nuclear membrane is perforated. Perforated means it has some pores, so materials can move in and out. And so that's what we call the nuclear pore. You understand the importance of nuclear pores when you talk about protein synthesis during chapter 2, protein synthesis. Then we have that's the nuclear envelope. It has a membrane, and the membrane, we have the inner membrane and outer membrane. So the nucleus is a double membrane organelle. So that is a distinguishing feature. A nucleus is a double membrane organelle with the inner and outer membrane. It has, that's the nuclear envelope. It has the nucleolus in the center, and then the nucleoplasm, which is like the cytoplasm of the nucleus. Coming to the micrograph, because we said we need to pay attention to this micrograph. If you'll see from this side, this one becomes our nucleolus, and then the openings here are what we call the nuclear membranes, as we are yet to see. But this cell is joined to another cell, so in the middle, it's joined to another cell. So that's a nucleus. So the pores in a membrane contains genetic information, which are the chromosomes, chromatins, DNA plus histone proteins. Histone proteins, DNA, chromatin, or chromosomes. Two meters of human DNA fits into nucleus, that is 0.000005 meters across. That's a fact. Now let us go to the powerhouse organelle number 2, which we call the mitochondrion. Mitochondrion is it's a powerhouse of the cell. So is the powerhouse of the cell. What is its structure? It is a sausage-like structure. That's number one. Sausage-like structure with the two membranes. The outer membrane is entire, and inner membrane is folded to form structures known as the crystal. The outer membrane is entire, the inner membrane is folded to form a structure known as the crystal that increases the surface area for formation or for aerobic respiration. So being a powerhouse means it's the source of energy. How does it create energy? It's because it contains what we call, it is a source, it contains, it is a source that's where aerobic respiration takes place. And if you look into this cell, you see it's on the sides of the cell. So that's the importance of the mitochondrion. Therefore, it's a powerhouse. Double membrane organelles. The outer membrane is entire and smooth. Inner membrane is folded. So this is when I ask you to describe the structure of a mitochondrion. So the description is basically in structure related to its function. And the same question is describe the adaptations. I'm interested in structure related to function. For example, I say that a mitochondrion is a powerhouse, sausage-like structure. It is double membrane. It's a double membranous organelle with the outer membrane smooth and entire, and the inner membrane is folded to form an internal structure known as the crystal. And the crystal increases the surface area for aerobic respiration or for enzymes that carry out aerobic respiration. However, shapes of these mitochondrions vary. So, a mitochondrion is a site for production of ATP during cellular respiration, which we call internal respiration, or which we may call tissue respiration. A mitochondrion have their own DNA, meaning that they can live independent of any other part. So, a mitochondrion and the cell are having what we call a symbiotic relationship. Symbiotic relationship means it lives in this cell to provide it with energy, and the cell keeps it to provide it with the support. Cell provides the support, mitochondrion produces energy. That's a symbiotic relationship between a mitochondrion and the a mitochondrion and the cell. So, meaning that a mitochondrion must have been an independent organism during times of evolution. But as life came up, it was engulfed into a bigger cell. And because it can live independent, so they began accepted to form what we call a symbiotic relationship. So we shall see more when we talk about the endosymbiotic theory. So large surface for cellular metabolism produces and contains its own ribosomes. So contains its own ribosomes and can reproduce independently. That's why we said it's like another living organism living in the other. So how do I describe the structure of a mitochondrion? I can use 4 to 5 statements to get my marks as required by the IB by any examination body. One, a mitochondrion is a sausage-like organelle. It is double membranous. The outer membrane is entire and smooth. The inner membrane is folded to form the cristae. It has its own DNA, meaning it can reproduce independently. It also contains the ribosomes of 70S. It is responsible for aerobic respiration. The inner membrane also contains a matrix, which is a fluid which contains enzymes. So don't forget about the matrix. That is the fluid that is encircled inside here, which contains enzymes that are responsible for carrying out this activity. If we are to see these mitochondrions, we can see one here. These ones are almost losing shape. That means they are becoming aged. They are becoming worn out. They are becoming aged. So worn out mitochondrions are result into they are important in the aging factor. So as they lose shape, the surface area for aerobic respiration reduces and the energy produced becomes less. So less energy is associated with the aging. So that's what we see with the mitochondrion. Then we look at another organelle which we call ribosomes. Ribosomes in eukaryotic cells, they have what we call, they are free. And if you look at them, they are bigger than the ones we saw in the prokaryotes. So these ones are 80S ribosomes. They are approximately 20 micrometers in diameter, larger than the ribosomes found in the prokaryotes. They have no membranes, and these appear as dark granules in the cytoplasm. So they appear as darker granules. You can see they make up of dysplasia. So all these dark granules here are ribosomes. So whenever you see ribosomes, just know you are talking about protein synthesis. So the role of ribosomes in any cell is protein synthesis. So when you find them attached to their endoplasmic reticula, they form what we call the rough endoplasmic reticulum. So they are responsible for assembling different amino acids during protein synthesis to form proteins. So the proteins formed by ribosomes are then sent to a structure known as the endoplasmic reticulum. Endoplasmic reticula consists of flattened membrane sacs, as we saw them in the mitochondrion. These flattened sacs, as you can see, this is a flat sac, flat sac, flat sac, flat sac, flat sacs, are called the cystini, and are often located near the nucleus. They are almost located near the nucleus. As you can see, actually, they mark the end of the nuclear membrane. And when they are attached with the ribosomes, they are referred to as the rough endoplasmic reticulum. And those extensions from the rough endoplasmic reticulum, but they don't have ribosomes, are what we call the smooth endoplasmic reticulum. So, the rough endoplasmic reticulum has ribosomes on them, and the smooth endoplasmic reticulum, they lack ribosomes on them. And that marks the differences. And even other differences come in function. I said whenever you see ribosomes attached, the first primary role is protein synthesis, assembling of different amino acids to form a protein. So, what does the rough endoplasmic reticulum does? It synthesizes proteins and transports them by the vesicle. So these smooth and rough endoplasmic reticulums, their role is to synthesize proteins, pack them, modify them. It modifies the proteins. Rough synthesis of proteins modifies them, giving them different structures or different roles they are going to serve, and then packs them. I repeat, synthesis of proteins, modification of proteins, and then packaging of proteins, and then transports them into a vesicle, which vesicle is now, it's like a pinch off. And those pinches are what we call the Golgi apparatus. So those pinches are transported by the Golgi. Now the Golgi is like the transporting mechanism. It's like the mover. It's the one that runs the errands of movement. So after synthesis, modification, and then packaging, the packaged protein is passed on to the to the vesicles on the Golgi apparatus before secreting them out of the cell. That's the function. So the pancreatic exocrine cells would have an abundance of these due to the constant production of digestive enzymes. So wherever you see production of enzymes, just know you have a lot of endoplasmic reticulum. Why? There is formation of of proteins, modifying them into enzymes, and then produce them out as enzymes. Then the smooth endoplasmic reticulum doesn't have ribosomes on it, as you can see here. And then what do? They're important in synthesis of lipids, and then packaging the lipids, and then modifying those lipids, and then passes them on to the Golgi apparatus. These exist in pancreatic cells. So these ones are found in pancreatic cells, then the pancreatic exocrine. So we have a lot of them in those functioning areas. Then we talk to the Golgi apparatus, which you mentioned as the transporter. Following this picture, I have a nucleus. And the nucleus is where we have the DNA. The DNA instructs which amino acids to be produced or activated for assembling to form a protein. After that is formed, assembling of these amino acids, which you call transcription or translation, takes place in this rough endoplasmic reticulum. Why does it take place here? Because here I have the ribosomes, which are responsible for assembling of amino acids. And after that, then the rough endoplasmic reticulum modifies those proteins formed, packages them, and then sends them off as the pinches here. So this pinch is transported. That's a vesicle containing already modified, packed a protein, whether it's an enzyme, whatever the case may be. And then after that, it is passed on to the Golgi apparatus. Golgi apparatus, or we call them the Golgi body, and the Golgi body now transports that packed protein, then sends it off wherever it wants to drop it off, from either from outside the cytoplasm to wherever. It will send it off, and when it reaches the point where it is required, it pinches, opens, and bursts, and then pours that enzyme, that protein, wherever it is needed. So what happens with the Golgi apparatus? It is the transporting part. So, like the endoplasmic reticulum, it is composed of flattened sacs called the cisternae. So now cisternae, we are seeing it for the third time. We saw it in the mitochondrion, where the inner membrane folds to form the cisternae that increases the surface area for the action of enzymes of aerobic respiration. Then we also saw cisternae in the rough endoplasmic reticulum and smooth endoplasmic reticulum, where it packages, modifies, and packages the proteins or lipids produced, and then passed them on to the Golgi body. The Golgi body also is an open sac that contains cisternae, and these cisternae, they don't have ribosomes, and is often near the plasma membrane. So the cisternae are shorter and more curved than those of our rough endoplasm or those of the endoplasmic reticulum. So the role of a Golgi body is to transport packaged materials. Can it be proteins? Let it be proteins or the other parts, which we call the lipids, and then drops them to where they are required. That's the role of the Golgi apparatus. In cell biology, Golgi apparatuses are almost in every syllabus. They ask much about the Golgi apparatus. They ask a lot about it. So better you take a caution when reading about it. So if you come here, you will see they are almost near the end of the cell. So there are these cisternae sac-like openings, which you see coming almost close to this. So in many cases, we ask this question here, and we ask for the function. There is assembling of amino acids, modification, packaging, and then pinching them off into a vesicle. Then a vesicle is attached to the Golgi apparatus. Golgi apparatus takes it and drops it wherever it wants to go. That's what happens. So Golgi collects, modifies, and distributes materials. It corrects as well. Sometimes also helps in packaging, modifies, and distribution of materials, the cell, especially the proteins. So that's what we see when we look at that. Now, if you look at the vesicles, a vesicle is a single membrane with a fluid inside, very small, used to transport materials outside the cell. So a vesicle is like when someone may have a swelling, and then you place it open, the fluids that come out. So the vesicle is also like that. When a cell has some materials it wants to eliminate, it will form a ball-like structure of a single membrane around those fluids, and then it gets close to the cell and then bursts open to pour those materials out of the cell. Then let's talk about lysosomes. These lysosomes are intracellular and arise from Golgi. They arise from the Golgi apparatus. They are spherical with a single membrane. So they stay dark in the micrographs due to high concentration of enzymes. So if this is a micrograph and it's drawn for you, we want you to explain what we see. And that's where you have the lysozymes. Why lysozymes contain enzymes, and these enzymes, this is where we have the hydrogen peroxide, which are responsible for detoxifications and production of did I say no? I said enzyme catalase. Sorry, that breaks the peroxisomes, which you call enzyme catalase, that detoxifies and breaks down hydrogen peroxide into water and the oxygen to reduce on the toxicity of the component once it develops inside the body. So cellular recycling. So these are this picture here tries to show us how the lysosomes and digestive enzymes they infusion the substance inside. That's the digestion. A materials inside, then digest the material. Then small molecules return into the cytosol, and then it comes here and pours them out. So then it comes back here and then reforms again, or the worn out organelles, get them. We get worn out organelles, get them digested by the lysosomes or break them down, fuses them, digests them. In lysosomes, there are always digestive enzymes to break down that we want to send out in a fluid form. Functions of the lysosomes, they contain digestive enzymes that break down ingested food material or damaged or worn out organelles inside. So lysosomes are what we call the suicidal sacs. Sometimes we call them suicidal sacs. Suicidal sacs. They are suicidal cell suicide. So they are the ones responsible for carrying out any kind of destruction of any cell, any organelle that appears to be worn out, that appears to be a problem. Because once they do, they come, engulf that cell, produce digestive enzymes. The digestive enzymes break it down into smaller components, then it comes out and pours them out. So they are called. So one of the questions: which one of the following are suicidal cells? Suicidal cells are what we call the the lysosomes. Suicidal. Uh, if we look at that, many diseases EGIT are caused by lysosomes or malfunctions, and then tie sacs are caused by lysosomes or malfunctions. That is when you don't have lysosomes functioning well, you develop some different complications. You can go and check much about the complications that may develop as a result of failure of having lysosomes break down break down the worn out cells or worn out materials. Then we look at this is the lysosome. It comes, it wants to engulf this food material. It will form a network around this. So this one looks like a white blood cell. Then it surrounds this microorganism, produces the bacteria, the digestive enzymes. They digest it down or break it down, and then later passes it on to the next generation. So this bacteria is about to be eaten by an immune system cell wall, spend the last minute of its existence within the lysosome. Yeah, that's what happens in immunity. Then vacuoles. What are vacuoles? Vacuoles are single membrane with the fluid inside. In plant cells, vacuoles are large and permanent, often occupying the majority of the cell volume. And in animal cells, vacuoles are smaller and temporary and are used for various reasons, e.g., to absorb food and digest it. So these are the vacuoles we see here. So what happens next? Vacuoles are important in storage of food, waste, toxins. Wastes increase surface area to volume ratio, allow plants to be rigid. So with the vacuole, they can store food, they can store wastes, excess water, and mineral salts. It also increases the surface area to volume ratio. And in the plants, vacuoles provide shape and rigidity of that cell. Then if you look at the cilia and flagella, everyone, if you touch your nose, you'll see you have some cilia, and other cells have flagella that are used for locomotion. For us, we have the cilia, This cilia is found in different parts. In the fallopian tube, we have the cilia in the fallopian tubes. We have the cilia in the respiratory system, and then we have the cilia, respiratory system. We have it in the reproductive system for yes. And what is the importance of the cilia? We also have the cilia on top. We have them in the ear. So what's the importance? They are tiny hair-like projections containing microtubules, and they are used to move the cell or move the fluids next to the cell. If you check inside your nose, if you sneeze, in the lungs, we have those cilia which beat up the mucus to take up against gravity and come in the nose, then you collect it here. So that's the importance of the cilia inside. Then in females, fallopian tubes, they beat up to propel the egg to go, and propulsion of the sperms to go for fertilization. So these are the cilia that we have in the lungs, as you can see. Cilia in the lungs. And then flagella, the thin projection from cell surface. They contain microtubules, are used for movement. Thin projections from the cellular surface contains microtubules used for movement. So, mature plant cells do not contain these structures, but some plant gametes do. Mature plant cells do not contain cilia, they don't contain flagella, but some plant gametes do. So, that's what we see. This can be a flagella. And basically, that's what we see. Microtubules and centrioles. Microtubules, yes. If you recall when I was talking about the differences between the flagella, microtubules and centrioles are responsible for what we call the cytoskeleton, which we call the cytoskeleton. A cytoskeleton, for a skeleton of the cell, for a cell to have a structure, it has a skeleton inside it. And those skeletons, it's where we have attachments, and those attachments are important in compartmentalization, whereby we shall have the nucleus held in the center, we have the mitochondrions on sides, we have the materials like Golgi apparatus carrying materials along. So we need to be very careful. These materials are very serious, actually very serious. So if you look at the microtubules, they are the ones responsible for the structure of every cell. So microtubules are small cylindrical fibers, are called microtubules, and they have a variety of functions. They are part of the structure of the flagella, and they play a role in cell division. You remember when a cell is dividing, we have centrioles, which move to opposite poles, and they start producing the microtubule or the spindofibers. Spindofibers are responsible for separating the sister chromatids or homologous chromosomes apart. So those cells with those features which are important in pulling the two to separate them from the kinetochore are what we call the spindofibers produced by the centrioles. Spindofibers produced by the centrioles. Then when we talk about centrioles, consists of two groups of 9 triplets. Are microtubules. They are mainly found in animal cells, not present in vascular plants or fungi. You remember when I said the flagella of a prokaryotic cell and the eukaryotic cells, we say they are different. I want you to look at this. I said that the flagella is made up of microtubules. And these microtubules are organized in the form of 2, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, 1, 2. So they are organized in groups of 2, 2 and make a circle. So I mentioned that circle is made up of 9 pairs of microtubules. 9 pairs of microtubules. Now, for for prokaryotic cells, they only consist of 9 pairs of microtubules. And if you see here, I have another pair, which would be the 10th pair, that is put in the middle. So that means this arrangement is a 9 plus 2 microtubule arrangement, which is typical of the eukaryotic cells. For prokaryotic cells, they have a 9 plus 0 microtubule arrangement. I repeat, microtubules are the ones responsible for forming some structures like those of the flagella. So the flagella in a prokaryotic cell and flagella in a eukaryotic cell, they are different in terms of the kind of microtubule arrangements that makes them up. They are all made up of microtubules, but the microtubule arrangement in a eukaryotic cell is a 9 plus 2 pair arrangement. Whereas in a prokaryotes, it's a 9 plus 0. So that means in prokaryotes, you don't see these 2. You will see this 1, 2, 1, 2, 9 times, but in the middle you will see nothing. For eukaryotes like these ones I'm discussing, we have a 9 plus 2 microtubules, which is typical of eukaryotic cells. So if we are to make a difference for microtubules, we shall say the microtubule arrangement of a prokaryotic cell is 9 plus 0, whereas in eukaryotic it's 9 plus 2 microtubule arrangement. Let's go to another organelle. This one also has its own different description. Describe the structure of a chloroplast, or describe how a chloroplast is adapted its function. If you look at a micrograph of this chloroplast, you will see it has stains, it has packs, which look like pancakes, as we can see, which you call the granum or granules. So how do we define a chloroplast? Description, introductory statement. A chloroplast is a double membrane organelle with the outer membrane and being entire and smooth. The inner membrane consists of a matrix, and it also consists of a stack of pancake-like structures, which are known as granules or grana, are joined together by thylakoids. It is a double membrane organelle. Outer membrane is entire, inner membrane contains a matrix, that is a fluid, which contains enzymes, mineral salts, and all that stuff. It also contains the fluid. The matrix inside is called the stroma. This fluid is called stroma, where light independent stage of photosynthesis takes place. You do much when you come to photosynthesis. It contains the stroma, that is the white colorless fluid you see, and then thylakoids, which are the intervascular thylakoids. That's the part that joins the stack of pancake-like structures, which we call the grana, from one to another, where light dependent stage of photosynthesis takes place. So the light dependent stage of photosynthesis takes place in these green parts, and the light-independent stage of photosynthesis takes place in the stroma. Well, the structure of this is it is it when you check it, it produces chlorophyll, a pigment that is responsible for the green color of a plant, or that traps sunlight for photosynthesis. So, can reproduce on its own because it has its own DNA as well. So, meaning this one is as a result of the endosymbiotic theory. There is a mutual understanding between the plant cell and the mitochondrion to live together. You stay, we benefit from each other. You stay in me and produce for me food, energy. I'll give you support. So a plant cell provides support. This one is responsible for breakdown of food, to release air, to break, formation of food. So it is 70, contains ribosomes, which are 70S, and inside are stacks of thylakoids, discs of flattened membrane, usually oval. So a chloroplast is in oval shape, and it's the site for photosynthesis or production of carbohydrates. When you check it, it has starch granules, may be present if produced lots of food. So those granules you can see, it has starch granules, whereas in animal cells we shall find glycogen granules. So that marks what we call a chloroplast, and it's only found in in plant cells alone. So think of a chloroplast as a solar panel of the plant cell. So that is what we see. Well, it's omnipresent, and algae have chloroplasts. Plant cells and algae are the ones that have chloroplasts. So if you look at it in this way, you will see this is a stack. Outer membrane, it's oval in shape. Outer membrane is entire and smooth. Inner membrane contains a fluid known as the stroma, and then it has flattened granules packed together to form a granum. And these granules are joined together by the thylakoids, or intervascular granum or thylakoids, and then these ones are the ones I'm talking about. You can call them stroma lamellae. So the fluid here is the stroma. This is the inner membrane. This is the outer membrane, and that's how it looks. It has its own DNA. It has its own ribosomes, so it can live independently. The granum flat sacs like solar panels. Why are these ones flat? They are acting as solar panels to increase the surface area for absorption, for production of carbohydrates, so glucose. Then the stroma fluid, it's the fluid that contains enzymes where chemical reactions take place. Then let's talk about cell wall. This cell wall here is very serious. When you go to university, you will see that it has a lot of information one would want you to explain. So the cell wall, it's basically found in plant cells. It's not in animal cell. And if you look at it, it has three regions. It has the outer part, plant cell wall structure. It has this middle part here where we have the proteins, which you call the pectin, and then the cross linkages, which you call the glycan, and then the cellulose microtubules. So you can see it has the plasma membranes. It has the primary cell wall, that's the part in the middle. A plasma membrane out, another plasma membrane. So in the middle here, we shall have these structures. It has proteins, which you call the pectin proteins, and then the cross-linking glycan, which you see in a yellow bead-like structure, and the cellulose microfibrils fibers. Cellulose microfibers. So these three joined together to form that strong wall. So its extra component known as an organelle, it's an extracellular component. It's not an organelle. It's out of the cell and secreted by all plant cells, also fungi and some protists. In plants, it contains mostly cellulose. Cellulose is a permeable, strong, and hard to digest little. So the cellulose, that is the cellulose microfibrils, it is permeable. It allows materials to go through it, but hard to digest. That's why, because of the cellulose, so this plant cell becomes a little hard and strong, provides rigidity. So the pectin beads, the cross-linked glycan, and the cellulose microfibrils, they provide that toughness of a plant's cell wall. That's why we say it is made up of cellulose. Unlike the cell wall found in bacteria, which is made up of peptidoglycan. So if you come here, we will see this is a chloroplast. This is a say vacuole, and then we can see this as a vacuole. Vacuoles keep on extending and reducing depending on the osmolarity of that cell and the seasons of the cell. So that's what we see. And this is the cell wall here that separates these two. Yeah. Peroxisomes, I've just talked about them. They are similar to ribosomes, except peroxisomes but bud off from the endoplasmic reticulum and have many roles and or involved in producing bile. So peroxisomes are enzymes. They produce bile. They are found much in the liver. They produce bile, fatty acid breakdown, cholesterol, myelin production, and getting rid of water. So they have many roles. One, are involved in producing bile. You know what bile is. Production of fatty acids from peroxisomes, a breakdown of cholesterols, those are peroxisomes, and myelin production, then getting rid of water. So they are also produced in the ever busy cells. So human cells contain several hundred peroxisomes, depicted in this photo as green spheres. So that's what we see. Extracellular matrix, that is the fluid, the matrix we've been talking about. We can have the intracellular matrix or extracellular matrix. That's what makes up the cytoplasm fluid like a part. In animal cells, are composed of collagen and glycoproteins, sugars and proteins. Form fibers. They form fibers. They anchor to the plant plasma membrane to strengthen it, so it provides the strength and support. It are also important in cell-to-cell interaction. That is extracellular matrix. Extracellular matrix. Yeah, cell-to-cell interaction. A matrix is a fluid part of it. Cell-to-cell interaction. A possible altering of gene expression. They can also alter gene expression. Leads to coordination of cell-to-cell. That's cell-to-cell communication or interaction. Interaction and communication. Extracellular matrices. Then may be involved in directing stem cells. That's also another function. And possibly cell migration and movement. So as you can see, we have the protoglycan molecules attaching here, protoglycan complex, protopolysaccharide molecules. You'll see cytoplasm, you'll see all that. So that's the extracellular matrices, a mixture of all these compartments. Well, I'll still go back to the cytoskeleton because I talked about it. The name is misleading. The cytoskeleton is the skeleton or the scaffolding of the cell, but it also like the muscular system as well, able to change the shape of cells in a flash. So that's the skeleton of the cell, made up of microtubules, microfilaments that provides the strength of this cell. That's why we call it the cytoskeleton. Yep. So then we can see a cytoskeleton gallery. What makes up a cytoskeleton? Uh, all these make up a cytoskeleton. So the fluids inside, the extracellular matrices, and then all those microtubules, they form a cytoskeleton. And macrophages, or using the skeleton to reach out for helpless bacteria, and the action. So centrosomes, usually a pair of centrioles, assemble microtubules for structure and movement, are involved in cell division, and our higher plants produce microtubules even though they do not have centrosomes. So these are the ones, centrosomes from centrioles produce these microtubules that are involved in cell division at all times of the day during cell division. So differences between a plant cell and animal cell. Plant cells have cell wall, animal cells don't have cell wall. Plant cells have chloroplasts, animal cells don't have chloroplasts. Plant cells have a large vacuole centrally located, animal cells don't have a large vacuole centrally located, but they are present and small. Plant cells store starch, animal cells store glycogen. In plant cells, they don't have centrosomes. Animal cells have centrosomes where we have those enzymes. Plant cells have a fixed plant angular shape. Animal cells are round, don't have, the shape is flexible. So that's where we see. Then we shall have to look at the electron microscope that have much higher resolution than the light microscope in our next video. Feel free to subscribe, invite a friend, and call them to go through. You can pause the lesson and then read. You can also pause and continue. You can pause and you write up something. You can always reach out to me and ask questions. After this, we shall be doing also questions regarding this area so that we can move as life goes on. Thank you for being part of the learning season today. I appreciate your patience. I appreciate your love and care as we continue to study. Always watch out for more biology lessons on this part. Thank you. Have a nice time.